Shown here is a nice figure which illustrates the structure of double-stranded DNA. After all, most of the DNA in our body is not single-stranded. Okay, it's usually double-stranded DNA that's complementarily base paired with associated complementary strand of DNA. So let's take a peek here. You'll notice that one strand of DNA is going to go from its 5' phosphate to its 3' hydroxyl group. What does that mean? Well, going back to the structure of that sugar, remember that there was a 1' carbon to which each nitrogen base was attached. There was a 2' carbon that dictated whether we were looking at RNA or DNA, depending on whether there was an oxygen there or not. And so the next carbon in that sugar is a 3', a 4', a 5', etc., etc. So what we mean by 5' phosphate is that attached to the 5' carbon of either the ribose or deoxyribose sugar is a phosphate group, a single phosphate group. Now, if you look at the backbone of DNA, there's a phosphate, a sugar, a phosphate, a sugar, a phosphate, a sugar, a phosphate, a sugar, etc., etc., etc. And at the very end of that linear strand of DNA, attached to the 3' carbon is going to be what? A hydroxyl group. For that reason, we say that DNA has a polarity. It goes from its 5' phosphate all the way down to its 3' hydroxyl group. That's the polarity of DNA, 5' phosphate to 3' hydroxyl group. All DNA will have that backbone. So what's going to make one individual's DNA different from another individual's DNA? It's going to be the sequence of the nitrogen bases. That's what's going to give us our specificity. So for example, this particular stretch of DNA is going to be read how? It will be read as TCAG. Okay, this particular stretch of DNA, for example. What is going to be holding the individual nucleotides together? So this would be a nucleotide. How do we know it's a nucleotide? It's a sugar, a nitrogen base, and a phosphate. Here's another nucleotide, a sugar, a nitrogen base, and a phosphate. What's going to hold these two together? It's a specific type of bond. Now, in biochemistry, for your board exam, there's only a few types of bonds you really have to worry about. You learn millions of them in organic chemistry, hundreds of them. But for our exam, there's really only a handful. This is one of the key ones right here, and it's called a phosphodiester bond. So a phosphodiester bond helps hold one nucleotide together with its neighboring nucleotide, this phosphodiester bond. So the properties now of double-stranded DNA. Even though the phosphodiester bond holds a single DNA's backbone together, and you have an associated complementary strand of DNA, what's going to hold these two strands of DNA together? And what holds them together are hydrogen bonds. So this gives us a property that we call complementarity. So DNA is always complementary. Does it say, hi, how are you doing? You look beautiful today. You look wonderful. You look nice. It's complementary, right? No, it's complementary, meaning that A's always base pair with T's, and G's always base pair with C's. Notice up here in the diagram, an A is always going to base pair with a T, a G is always going to base pair with a C. That rule is going to be universal among double-stranded DNA. Because it's universal, you universally have to remember it. The other property here is that DNA is anti-parallel. What does that mean, anti-parallel? Well, if DNA were parallel, then you would imagine that the 5' phosphate at one end of the double-stranded DNA would base pair with the 5' phosphate on the complementary DNA strand. But is that what we see? No, it's not what we see. What we see is that the 5' phosphate on one strand of DNA is going to base pair with the 3' hydroxyl group end of the complementary DNA strand. So for that reason, we say it's anti-parallel. The 5' end of one strand is associated with the 3' end of the complementary strand, Whereas at the bottom, the 3' end of one strand is associated with the 5' end of the complementary strand. So DNA is anti-parallel. Why are we taking the time to mention this? It's because it's absolutely critical when it comes to replicating DNA, when it comes to transcribing DNA. Those processes are always going to happen in a complementary, anti-parallel fashion. This theme will come back again and again and again as we review replication and also transcription. Another point I would like to make about this is you see that A's and T's form how many hydrogen bonds to hold themselves together? They form 2. What about G's and C's? Well, G's and C's are eventually going to, even though not quite shown here, are going to form 3 hydrogen bonds to hold themselves together. You see it better down here in this particular illustration. G's and C's form 3 hydrogen bonds. So, what is a stronger bond? An AT bond or a GC bond? Well, it's going to take more energy to break the GC bond because there's 3 hydrogen bonds. It's going to take less energy to break the AT bonds because there's only 2 hydrogen bonds. Keep that point in mind because it's possible you would be asked a question about comparing different types of DNA and asked which one would require a higher melting temperature, i.e., which DNA would require more energy to break the hydrogen bonds to make the DNA single-stranded. Well, clearly, the DNA that would take the most energy to become single-stranded or become melted would be the DNA that has the most GC bonds in it, or the DNA that simply has the most G's and C's. Which DNA would take the least amount of energy to break? It would be the DNA that has the most A's and T's present. Because remember, when A's base pair with T's, there's only 2 hydrogen bonds. This is a topic that's been tested, and so I think it's worth your while to remember that GC bonds are more energetic than AT bonds. When we read a sequence, when we as humans read a sequence of DNA, how do we do so? By convention, when we read a DNA sequence, we typically do so from 5' to 3'. That's just the convention. That's the way it is. Okay, in English, the English language, we usually read from left to right. Okay, you don't usually read from right to left, unless we're trying to read upside down. But in DNA, by convention, we read DNA from the 5' to the 3' end. So, for this particular strand of DNA on the left, how would this read? Remember, we're going to start at the 5' phosphate and go down to the 3' hydroxyl. So this would read TCAG. Okay, so from 5' to 3', that strand would read TCAG. There's another way that a test question could present DNA sequence to you, and that is to put these little P's in here. And what do those P's represent? They simply represent phosphate groups. So this would represent the phosphates that help hold these DNA nitrogen bases together, the phosphodiester backbone, phosphate sugar backbone. So don't get tripped up by these P's, and that's probably not even going to be asked of you. But just on the off chance it is, I want you to be familiar with seeing it that way. This is where some folks reviewing DNA information get tripped up, and I want to make sure that you're very clear on how this works. The sequence here from 5' to 3' reads TCAG. So from 5 to 3' it reads TCAG. But how does it read from 3 to 5'? If you start at the 3' end, it reads GACT. Notice down here, from 3 to 5', GACT. Aha! Is this sequence different than this sequence up here? The answer is no, it's not. And you need to realize that in order to comfortably understand this. You could write the sequence from 5 to 3', TCAG. Or you could simply write this sequence from 3 to 5 prime, and it would read GACT. And that's what's been done. So whether the test questions present the information to you from the 5 to 3 prime or the 3 to 5 prime end, it doesn't matter. You need to be familiar with both orientations. Now, remember, we as humans tend to read DNA sequence from 5 to 3 prime. That's going to be a lot different than the RNA and DNA polymerases because they're actually going to read the DNA from 3 prime to 5 prime. That will have more relevance when we get into that particular subject. So, shown here is something called Chargaff's rules. In double-stranded DNA, and actually this was a particular set of rules which was defined by a scientist named Erwin Chargaff some 100 years ago, and he was able to decipher that the amount of A's and the amount of T's were equal. The amount of G's and the amount of C's were equal. And furthermore, the number of purines and the number of pyrimidines within a DNA species were always equal. And actually using these building blocks, Watson and Crick were able to sort of decipher the structure, to solve the structure of DNA back in 1953. So how would this be applied to a test question? Because that's what we're after, right? Well, using Chargaff's rules, you can be asked to figure out what percentage of one nitrogen base is present if you're given the percentage of another nitrogen base. Let's look at this example. So, if we're given a sample of DNA which has 10% G's, how could we calculate the percentage of T's? Well, let's use Chargaff's rules. Remember that the percentage of G's is always equal to the percentage of C's. So, if you're given a sample with 10% G's, what percent of that sample is C? It's going to be the same, 10%. So now, if 10% is G and 10% is C, that together equals 20%. That equals 20%. So what's left over is 80%. We know that the percentage of A equals the percentage of T, so to figure out the percentage of T, all we have to do is divide that in half. So 80% divided by 2 would give you 40%. That should be the percentage of T, and absolutely that is. Let's go through that one more time. If you're given that the percentage is G, you can say safely that the percentage of C is the same, so it would also be 10%. So 10% plus 10% gives you 20%. What's left over, i.e. the A's and T's, must be equal to 80%. It has to be because you can't go over 100% here. So the percentage of A's and T's together must equal 80%. Because A's and T's are equal, you could divide this value in half to give you 40%. That's how you would approach answering one of these questions that utilizes Chargaff's rules. Let's practice one more. Suppose a sample of double-stranded DNA has 35% A. What is the percentage of C? Okay, how do we figure this out? What's the first step? If there's 35% A, where do you go next? Well, where you go next is to figure out how much T is there. If there's 35% A, well, then there must be 35% T. 35% plus 35% gives you what? 70%. What's left over? Well, 100 minus 70 is 30%. So 30% of the DNA bases are G's and C's. So how do you figure out the C's? You just divide it in half. Okay, so 30 divided by 2 would be 15%. And that's your answer, 15%. Once again, if 35% are A, then also you can surmise that 35% are T. Together, 35 plus 35% is 70%. So what's left over? 100 minus 70 is 30%. 30% are G's and C's, and G's equal C's, according to Chargaff's rules over here. So if you divide that in half, you're going to get 15%. If you're the type of person that wants a really good shortcut, and I think shortcuts are great, If you're given one of these Chargas rules questions, and you're given the percentage and composition of one of the nucleotide bases, you simply multiply that by 2 to give you the G's, C's, or A's, T's. You then subtract that percentage from 100, and then you divide that by 2. That would be the easy shortcut for answering one of these questions. You multiply it by 2, you subtract it from 100, and then you simply divide it by 2. What happens if they give you a curveball? Okay, they're going to give you a curveball question. They're going to say, suppose a sample of DNA has 10% A and 50% G. What is its significance? So here you are, you listened to this review, and you found this wonderful shortcut, and you said, okay, I'm going to take 10%, I'm going to multiply it times 2, I get 20%, I'm going to subtract that from 100, I get 80%, I'm going to divide by 2, I get 40. What? Huh? What? This is fit. Wait, does this make sense? Oh my God, the rules of the universe are falling apart on me. What am I going to do? Relax, okay? There's a specific example when Chargas rules don't apply, okay? Notice up here, this says in double-stranded DNA, or even, for that matter, double-stranded RNA. It's double-stranded DNA. So if you see an example where Chargas rules are going out the window, the significance is that you're probably looking at single-stranded nucleic acid. Now, where would you see single-stranded nucleic acid? Well, there's certain viruses that like to invade your body that are single-stranded nucleic acids. So if you were given a situation where Chargas rules don't seemingly apply, chances are it's probably some sort of virus that's a pathogen that's infecting your body. Shown on this slide is the prototypical ribbon structure of DNA. You see that the sugar phosphate backbone is represented by the ribbons on this particular figure, and the inside of this are the associated hydrogen-bonded nitrogen bases. So another way to think of this is as a ladder that's been twisted around itself. So that puts DNA into the configuration of a double helix. The form of DNA that's present in most of your bodies, in the cells of most of your bodies, is the B form of DNA. We don't really have to worry about this much more than just remembering the B form of DNA. It actually forms a right-handed helix. There are other forms of DNA. There's even a left-handed helical form of DNA that would be like this. That's the Z form of DNA. We don't have to worry about it. If you're ever asked a question about the structure of DNA, the form that predominates in your cells, it's the B form of DNA. In this form, obviously, the sugar phosphates are on the outside, whereas on the insides are the nitrogen bases, hydrogen bonded with one another. Because DNA forms a double helix, this gives an additional structural motif to the molecule, so that you can see that the DNA is divided up into a major groove and a minor groove. This doesn't have too much relevance for your studies, other than for you to understand that certain drugs, certain DNA molecules are going to bind to either the major or the minor groove, and that's going to give them access to the DNA to perhaps intercalate or interrupt the DNA structure. For each turn of the DNA molecule, there's about 10 bases. So each time the helix goes around one time, there should be about 10 bases present there. What's the significance of that for you? If you're given a question about some newly discovered DNA structure, and let's say that there's 12, 13, 14 bases per turn of the helix, how does that look compared to normal DNA? Well, it's squeezed in. It's DNA that's squeezed in a little bit tighter than normal. So it's more highly packed or tightly packed DNA. If the DNA has less than 10 bases per turn, then it's going to be more relaxed. So there's going to be enzymes that cause DNA to be packed closer and enzymes that cause it to relax. This would make it more than 10 or less than 10 bases per turn. And in fact, the bases that are stacked together form, in addition to the hydrogen bond that occurs between the nitrogen bases, there's hydrophobic interactions that occur among adjacent nitrogen bases. So these hydrophobic interactions provide additional stability. So it's going to take a good amount of energy to cause the DNA to come apart. The coming apart of DNA is what's depicted on this next figure, and that's better known as DNA denaturation. So what is denaturation? Let's define this. It's taking double-stranded DNA, somehow heating it, or through an enzymatic step, causing the cleavage of the hydrogen bonds and hydrophobic interactions that hold the strands together. So cleavage of these bonds, either through heat or catalytic activity, is going to separate the strands. So now you have two single-stranded DNA entities. Why is this important? Well, it's important because later on, when we talk about PCR, which is polymerase chain reaction and adding primers in, or we talk about southern blotting and hybridization or northern blotting and hybridization, well, these techniques rely on the fact that the DNA becomes separated, but if you allow it to cool down, it can then renature. Another word that you can put in here if you choose is annealing. So renaturation or annealing of DNA is when the two strands come back together. If you pictured mixing in a new strand of DNA prior to allowing this to cool down and renature, well, one of the new strands could bind to one of these existing strands of DNA that have been separated initially. And that's actually how PCR priming works, and that's how southern blotting and hybridization works. The two strands are separated, a new shorter piece of DNA is allowed to adhere to that, it's cooled down, and it can stick to it through complementary base pairing. We'll review that a little bit later on.